And we're going to spend some time with Dr. Christine Cooper, who has research and teaching academic position as a senior lecturer at the School of Molecular and Life Sciences at Curtin University in Western Australia. Her research interests are primarily the thermal, energy, and water balance in terrestrial vertebrates, while looking at how things like evolution, body mass, life history, and environmental factors can influence those traits. And she gets to work with what I think are the coolest group of mammals, the monotremes, the echidna, the platypus. So let's bring her in now. Let's get into this story. Here we go. Hi, Christine. How are you?
Hello, good, thank you.
Good, good. It is so great to have you joining us today. I know it's probably about supper time there for you in Australia, but it is great to see you, and I'm excited to get into the echidna world.
Thank you very much for having me, and it's always a pleasure to talk about echidnas.
Excellent. Well, I am going to bring in your presentation, such a cool picture, and I'm going to let you take over for a little bit.
Okay, thank you. So I'm going to talk tonight about echidna physiology and how they are coping with an changing environment. So short-beak echidnas are the most widely distributed Australian mammal, and they're really special because they're monotremes, which means they're really unusual amongst mammals in that they lay eggs. And they're found all over the Australian continent, from Tasmania all the way up through Australia and even into New Guinea. And they're special as well because they haven't suffered the same level of decline that we've seen in a lot of other Australian mammals, in particular some of the marsupial species. And part of that reason is because they have a protective armour of spines, and that deters a lot of the introduced predators like cats and foxes that have caused havoc amongst a lot of our native species. But as well as having a spiny armour, they also have a really successful physiological strategy that has enabled them to cope with living over such a wide climatic distribution, and appears to be going to hold them in good stead into the future with climate change. So echidnas have always been thought to be poor thermoregulators. So a lot of the early studies of echidnas concluded that they weren't very good at maintaining their body temperature. So echidnas can't sweat. They don't have sweat glands like we do. And so when they get hot, sweating's not an option for them to lose heat. They also can't pant. So they can't lose heat like a dog will by panting. They don't have vasomotor adjustments, so they can't change blood flow, supposedly, to different areas of their body, which means they're quite intolerant to high temperatures. And some of the early studies suggested that echidnas can't tolerate temperatures over 35 degrees Celsius and that they will die at temperatures above that. Now, that's really unusual considering their broad geographic distribution, and we had suspicions that they're probably more thermally tolerant than what some of these early studies suggested. But the idea was that echidnas can't physiologically cope with high temperature, and they have to become inactive and rely on behavioural mechanisms to avoid these high temperatures. And indeed, our data show that they don't like to be active when it's hot. So here's some echidna activity data in spring and moderate temperatures, and they're active mostly at night, but also through the daytime. However, in summertime, when it gets much hotter, they're completely inactive through the day, and we're only seeing activity here in the evening. And they shelter in places like caves and burrows and hollow logs to try and avoid those high temperatures. But we've observed echidnas out and about, active at temperatures up to about 38 degrees. And the highest temperature we know that an echidna has been exposed to and survived was nearly 42 degrees. So even though the early studies suggest they die above 35, we've got pretty good evidence that they don't. So the question is, how do they do this? If they can't pant or sweat, what do they do? Well, some of the work that we've done in the lab shows that they can increase their evaporation at high temperature. And by increasing their evaporative water loss, they increase their heat loss. They can also increase their thermal conductance at high temperature. So thermal conductance is how easily an animal can lose heat. So when it gets hot, echidnas can increase their heat loss to the environment. But the great big question is, how do they do that? And we know they can increase the permeability of their skin surface to water to increase their evaporation, but we had some other ideas about how they might do this. We thought they might use thermal windows or areas of their body that they could use to dump heat. And my PhD student, Justine, observed echidnas blowing bubbles at high temperatures in the lab. So we thought that perhaps they might use blowing bubbles from their nose as a way to increase their evaporative water loss. So we set out to film echidnas in the field. So we used a thermal imaging camera, and we would drive around the tracks at my study site, which is Drandra Woodland, about 200 kilometres east of Perth. And when we saw an echidna, we would stop and we would film this echidna with our thermal imaging camera. And that allowed us to measure the surface temperature of the echidna. And we could measure surface temperature of all different parts of their body as they went about their foraging and other activities in the environment. And we monitored echidnas for every month of the year for several days, and we were able to film over 80 echidnas. And we collected data between 10 in the morning and about 11 o'clock at night. And we found that echidnas were active over a wide range of temperatures, including temperatures that well and truly exceed the temperatures where they're supposed to die. And these are echidnas behaving in the wild, coming out and being active and being quite happy and healthy and clearly not dying at environmental temperatures. And that's what black globe temperature is a measure of. It's a measure of ambient and other aspects of temperature, including solar radiation, up to about 47 degrees. So clearly, they're much more tolerant of high temperatures than what some of those early studies suggested. So using our thermal imaging videos, we then were able to measure the temperature of different body parts. So this is their body temperature for an echidna that's resting at a comfortable environmental temperature. And if we measure the surface temperature of the ear, that's pretty closely approximates body temperature. So we can use ear temperature as an indication of body temperature. The ventral surface, the inside the legs and the middle of their spines are all quite hot, but their nose base in their body is much cooler, and their nose tip is even cooler than that. And that gave us some ideas of how they might be dealing with heat exchange. So the first thing we notice is their spines provide them with really good insulation. So their spines act as a cover, and when it's cool, they can lay those spines down and they can stop their body heat escaping. And they can also act as a barrier to gaining solar heat. So they provide them from insulation, stopping them gain too much heat from their environment and also stopping them lose body heat when it is cool. But the spines are also flexible. They can stand the spines on end and increase the heat loss, or lay them down and keep the heat in. So they give them really good insulation, but that insulation is very flexible. And we can see this if we look at ear temperature. So this is the temperature of the ear, and this is the environmental temperature. And ear temperature is reasonably different from environmental temperature because it's quite influenced by their body temperature. However, if we look at the surface of their body where their spines are sitting, we see that that much more closely approximates the environmental temperature. And that's because the spines are acting as an insulator. The top of the spines is closer to environmental temperature and the bottom is closer to body temperature. It's stopping them exchanging heat with their environment. The next thing we were interested in is how they might be using evaporative windows. So the best known example of an evaporative window is what kangaroos do. They lick their forearms when it gets really hot. And if we look underneath the skin on the forearms, they've got lots and lots of blood vessels close to the surface. So when they lick their arms, the water evaporates and it cools the blood underlying in the tissue beneath. And it's just like us running our wrists under cold water on a really hot day. So that's what we would call an evaporative window. Well, echidnas have a big blood sinus on the tip of their nose. And Justine noticed in her experiments that echidnas would often blow bubbles at high temperatures. The bubbles would burst and would wet the tip of the nose. And the big blood sinus sits underneath that moist area. So we wondered if that perhaps could be a way that they're keeping cool. So when we thermally imaged our echidnas, we found that sometimes in the field they would be blowing bubbles. And when those bubbles burst over the nose would become wet. And because that water would then evaporate, it would cool the nose. So echidnas do have really cold noses. Now, it's important that they keep their noses moist for electroreception. That's how they find their prey. But that moisture also allows them to dissipate heat when it's hot. And so their noses stay cold, cooling the underlying blood, and it works as an evaporative window, just like the kangaroos licking their arms. We also found that echidnas have what we call thermal windows. They have parts of the body they can use to dissipate their heat. So if we look at this echidna here walking away, he's been sitting on the cool ground and they don't have spines on their ventral surface. So if he presses that against the cool ground, you can see he's left behind a heat patch here. So he's able to dump body heat by transferring that heat by conduction to the ground surface. They also have bare patches on the inside of their legs and between their spines. And so if they erect their spines or expose their legs, it means they can lose heat by convection to their environment. So these areas make really good thermal windows because they can open them by exposing them and lose heat. Or if it's cool and they don't want to lose heat, they can tuck their legs up or they can lay their spines down and close those thermal windows. So thermal windows, evaporative windows, and well-insulating spines give echidnas really good ways to help them cope in a hot environment. Echidnas are also quite variable with their respect to their physiology. So if we compare the physiology of echidnas here from Western Australia, where it's typically quite hot and dry, with echidnas from Tasmania, we can see a whole lot of anatomical differences. The most obvious is that Tasmanian echidnas have fewer spines and a lot more fur compared to the really spiny and not at all furry West Australian echidnas. And this is also related to physiological differences. So Tasmanian echidnas have much higher energy use. So their metabolic rate is higher. That means their heat production is higher and therefore they have a higher body temperature. So in a cold, moist environment where there's probably a bit more energy and food around, Tasmanian echidnas produce more heat and can keep themselves warm. West Australian echidnas have lower water loss than Tasmanian echidnas. So West Australian echidnas live in a hotter, drier environment and they need to conserve water. So in general, their evaporative water loss is lower than what we see in Tasmanian echidnas. Echidnas are also really flexible with their body temperature. So here's some data we collected for West Australian echidnas. So the black line here is the echidna's body temperature. The dark grey line is a temperature logger glued onto its back, and the pale grey line is the ambient temperature. So you can see the echidna's body temperature varies throughout the day, and their body temperature ranges from about 11 degrees up to about 36 degrees. So they're hugely variable, and that means at nighttime when it's cool, they just let their body temperature drop and they save all the energy they would need otherwise to keep warm. And then they warm up during when they're active and through the day, and then let their temperature drop again at night. Echidnas can also drop into a period of torpor, so they let their body temperature get really, really low and they become inactive and just rest. And sometimes they'll actually go into multi-day torpor or hibernation. So they'll allow their body temperature to drop down and they stay cold and inactive for several days. And they tend to do this on days where the weather is really inclement, so it might be raining and cold. You can see here the weather's not very nice. We don't get these nice sunny daytime peaks, and the echidna enters hibernation. Now in Western Australia, where it tends to be nice and warm and sunny most of the time, the longest we found our echidnas to hibernate was about 10 days. But in Tasmania, where it's much, much colder, echidnas can hibernate for months. So here's body temperature in the solid lines, is ambient temperature with the dots, and this echidna is hibernating over time. And they have these little periods of arousal for a day or two throughout the hibernation period, and we don't really know why they do that. We think it might be something to do with water balance, that maybe they need to drink. It might be something to do with sleep, because when animals are hibernating, they're not actually sleeping properly, and it might be necessary for them to reform some of their memories. But we don't really know why they arouse. We just know that they have to do that. But they stay inactive and they're pretty much hibernating for months at a time, whereas our WA echidnas only hibernate for a few days if the weather's bad. And that means echidnas can avoid inclement conditions, and they've got flexibility to do that depending on what their environment is like. And this ability to hibernate and become inactive is really important if there's some sort of natural disaster, like a storm or a flood, or in this case, a fire, that allows them to avoid those situations. So we were really fortunate that the national park that I work in, the staff there had to do a prescribed burn. So a lot of Australian plants need fire for the seeds to germinate, and therefore they have to burn areas of the park on a regular basis to allow that seed bank to germinate. So we knew that this prescribed burn was going to happen, so we went out and caught some echidnas in the area that was going to be burnt, and we put some GPS trackers and radio trackers and body temperature loggers on them, and we also got some control echidnas from an area that wasn't burnt. We then let them go. The staff did their burn, and we went back to see what happened to our echidnas during the fire. And we found that after the fire, the echidnas didn't really change where they lived, even though their environment went from this beautiful woodland environment to this fairly desolate place where it was going to be really hard for them to find shelter or food. They didn't move somewhere else. They pretty much stayed in the same place where they were before the fire. But what they did do is they became much more inactive. So if we look at how much time the echidnas were spending in multi-day torpor before the burn compared to after the burn, the echidnas in the burn area increased the time they were spending inactive much more than our control echidnas. So before the burn, they were pretty similar. After the burn, we still get a little bit of hibernation happening, but our burn group become much more variable in terms of how low their body temperature gets and how long they stay in hibernation. So this suggests that being able to drop your body temperature down, hide away and become inactive is really valuable for surviving natural disasters. And in fact, being able to use torpor is one hypothesis as to how some of the early mammals were able to survive the meteorite impact that was responsible for the extinction of the dinosaurs. So a flexible body temperature is really important for survival. So why are we so interested in how echidnas survive and how they might cope in a future warmer environment? Well, echidnas have a really important role in the ecosystem. They dig a lot, and this digging has important ecosystem function in that it turns over the soil and it supports plant growth. So we were interested in looking at how much and over how wide an area do echidnas dig. So we fashioned some little backpacks for echidnas that consisted of a GPS and an accelerometer, a bit like an echidna Fitbit. It had a temperature logger and a light sensor, and we glued those to the echidnas. It's great when you work on an animal with spines because you can glue things to their back. And then we let the echidnas go, and we'll be able to collect data about what they were doing and where they were in their environment. So we found that most of the time echidnas sleep, so they're inactive for the majority of the time at any time of the year. But when they are active, most of the active time is spent digging and foraging. And that's important because they're digging over quite a big area. So this graph here shows the relationship between body mass, the size of the animal, and how far they move around. And different animals move around a different amount. So herbivores don't move that much. Carnivores move around a lot more because they've got to find prey. It's not as abundant as it is for herbivores, so they've got to travel over a bigger area and they get lots of energy for their food. But we were really surprised at how much echidnas move. This is sort of the average line for mammals, and echidnas are right up here. So they're moving over big distances, and when they're moving, they're foraging. So here's a GPS track of our echidna. This is where he's moving in the landscape, and this is when he is digging and foraging. So blue when he's walking and red bits when he's digging and foraging. So he's digging over quite a big area in the landscape and travelling over a large area, which means his ecosystem services are being applied to quite a large part of the environment. So this is really important because they dig, and they dig quite a lot. This is a typical echidna digging trench here. What they're doing is they're breaking up the soil, they're loosening it up, they're allowing water to infiltrate into the soil better, they're burrowing leaf litter and other nutrients, they're essentially planting seeds, they're burying seeds into those digging pits, and so they're making the soil much more heterogeneous, they're making it less hygroscopic, they're promoting plant growth. And the echidna's role in this is particularly important because lots of the other marsupials that used to do this job have become really rare or even extinct over much of the Australian continent, particularly in the arid areas. So the echidna, with its wide distribution and its ability to withstand a lot of the pressure of introduced predators, is able to survive and carry out this role throughout much of Australia, where other marsupial mammals used to do this job instead. So it's really important that echidnas are preserved in their environment and that they can carry on to perform this really important ecosystem function. And a lot of the early work that suggested echidnas were very poor thermoregulators meant that it was probably not very good news for echidnas moving forward with climate change. But our work has shown that echidnas are much more tolerant of heat and aridity than what we used to think, and therefore we have some hope that they will be able to persist in most of their range in a changing climate.
All right. Christine, thank you so much. I love that we're ending on that image as well. It's such a cool echidna image, that thermal picture of what we've got going there. Just, yeah, very, very cool. And so, what I like about this story is it's proving something that we thought was right in science. And as it tends to happen in science, is as we learn more, we find out new things, and that what we believed before isn't true. And then I think it's a really interesting example of how a species that maybe we thought would be really impacted by climate change does have that little kind of built-in adaptation that's going to help it a little bit.
Yes, it's a really nice good news story. So, I mean, they're still not amazingly tolerant of high temperatures, but they're certainly more tolerant than what we used to think they were. And that is certainly good news for at least a lot of the continent that they're living over.
All right. So we got a question here about field work. Can you take us into the field? What's your field work look like?
I'm really fortunate to have Dryandra as a study site. Dryandra is open woodland. It's really close to Perth, so it's only about 200 kilometres from Perth, which by West Australian standards is really close. And it's very open woodland. It's got a lot of access tracks. So the way you find echidnas is you drive around at about 5 kilometres an hour and you just look for them. And then when you see one, then you either film it or jump out and grab it, or depending on what your approach is. So it's a fantastic place. And echidnas are actually really hard to find because they're incredibly cryptic. You don't see them very often, and there's nowhere else I know that you can go to and reliably find an echidna. So I can go to Dryandra, and I think I've been working there for about 25 years, and I think I've only had 3 or 4 trips where I haven't seen an echidna. So it's a fantastic place to study this really special mammal.
Yeah, no question. And so, in the career path that you have, you've been able to study other mammal species as well. Can you give us an example of another species that you've studied?
Yeah, I'm a comparative physiologist, and one of the really nice things about that is I ask questions about how animals solve common problems. And because a lot of that, the answers to that comes by comparing different species, it means I get to work on a whole range of species. So most of my work has been on birds and mammals, but I've worked on over 60 species of marsupials, a whole lot of different birds, including parrots in particular. I really enjoy working on parrots, monotremes as echidnas. But probably my favourite animal is the numbat. I was really fortunate to study numbats for my PhD, and they're a really special marsupial, which also eats termites as their diet. So I sort of have ended up working on animals that eat ants and termites.
Yeah, they've got that super cool tongue similar to the echidna that can come out and snag those tasty morsels. So to wrap up the field, right? When you're out in the field, it's just the law, I think, that things don't always go as planned. So what are some of the challenges that you faced with this research when you were out in the field?
Probably the hardest thing is the climatic conditions. So, in summer out at Dryandra, you might get days of 46 degrees. So sometimes it can be really challenging. Echidnas are not always the most cooperative animals, so they're actually incredibly strong. And they learn pretty quickly that when they hear us coming, that if they get in a hollow log or in a cave, we can't extract them. So I have had some nights in summer where it's been really hot and they just don't come out. And it's been 1 o'clock in the morning and I've been really desperate to get my equipment back, and I've been chasing this echidna, trying to extract it from a cave, and getting a little bit frustrated because I really wanted my data back that was on the loggers that was on it. So yeah, sometimes uncooperative subjects can be a little bit frustrating, but we're pretty lucky to be able to work on them, so that's not too bad.
All right, fair enough. Well, Christine, thank you so much for taking us into this really cool research. It really just kind of hammers home that we are just scratching the surface of what we know about our ecosystems and the animals and the species found within. We have so much work to do to better understand the world around us. So thank you for filling in another really cool piece for us.
Thank you very much for having me and for providing the opportunity to chat about echidnas.
All right, excellent. Well, enjoy your evening. I hope you can tune into a few more events, and I have no doubt that we'll connect again soon.
Thank you very much.
All right, thanks, Christine.
See you later.